Assalamu alaikum. In our previous video, we talked about the specific immunity. In this video, we are going to have an overview of the specific immunity, that how the specific immunity works and what are the procedures or the cells that are involved in the specific immunity. So there are two arms of specific immunity. One is known as cell mediated immunity and the other is known as antibody mediated immunity. In the cell-mediated immunity, as the name implies, some cells are involved that are producing an immune response. And in antibody-mediated immunity, antibodies are produced. So the cells that are involved in the specific immunity include the APCs, which are the antigen-presenting cells. These cells include macrophages, dendritic cells, and B cells. Next, we have T cells. T cells are further categorized into two types on the basis of specific types of receptors that are present on their surface. There are some T cells which are known as CD4 cells. Those have a receptor on their surface which is called CD4, and those cells are called helper T cells. Then the other type of T cells which have a receptor CD8 on their surface, they are known as cytotoxic T cells. And the other type of lymphocytes, B cells, are also involved in the specific immunity, and B cells are involved in the production of antibodies. In our video of inflammation and phagocytosis, we talked about that the pathogens were present at the site of infection, and the macrophages engulfed those pathogens at the site of infection and then phagocytosed them and killed them. Those pathogens were phagocytosed at that site of infection in a phagosome. Then the phagosomes were fused with the lysosomes, and then the lysosomal enzymes disintegrated bacteria and killed them. So now the macrophages will take the antigens that were present on the bacterial cell. They will take those antigens and will present those antigens on their surface. So that's why they are called as antigen presenting cells. They will present those antigens on their surface and then they will take those antigens to the helper T cells. But helper T cells cannot recognize antigens as long as some other molecules are not linked with those antigens. Those molecules that are linked with the antigens are called as major histocompatibility complexes. Major histocompatibility complex is a protein that is encoded by the antigen presenting cell along with the antigens are presented along with the MHC molecule on the surface of APC. There are three types of major histocompatibility complexes. There is a type 1, type 2 and type 3. Type 1 major histocompatibility complex is involved in viral infections, or we can say that they are involved in the presentation of intracellular antigens. And the MHC class 2 major histocompatibility complex type 2 is involved in the presentation of extracellular pathogens such as bacteria. Now the antigen presenting cells have MHC class 2 molecule on their surface along with the antigen. They will move toward the helper T cell or CD4 T cells. The CD4 T cells have two types of receptors on their surface that will recognize this antigen. First is the TCR, which is called as T cell receptor. This receptor will recognize the antigen, but antigen is not enough to activate the helper T cell. So the receptor CD4 will recognize the type of major histocompatibility complex along with the antigen. So now the helper T cell has recognized the major histocompatibility complex and also the antigen. Now to make this interaction more prominent and to further activate the helper T cell, the antigen presenting cell will produce another type of chemical, and this chemical is known as cytokine. And the specific type of cytokine that will be produced at this stage by the APC is known as interleukin 1. Interleukin 1 will further activate the helper T cell. Now the helper T cell is activated. Now the activated helper T cell will produce another type of molecule. The interleukin 1 that was produced by APC will bind to a specific receptor on the surface of the helper T cell, and that binding to the receptor will produce a signaling pathway within the helper T cell, and that signaling pathway will go and further activate some genes, and those genes will produce two types of products. One is product is the interleukin 2, and the other product is the receptor for interleukin 2. So it is an example of autocrine signaling, in which a cell is producing a signal and is activated by that signal. Now the interleukin 2 is produced by the helper T cell, and interleukin 2 receptor is also present on the surface of the helper T cell. Then interleukin 2 will bind with the interleukin 2 receptor, and now the helper T cell is activated and it can further perform two functions. First function is that it can go and activate the cytotoxic T cell, or which is also known as CD8 T cell. How can it activate that cell? CD8 also has interleukin 2 receptors on its surface. The interleukin 2 that is produced by the helper T cell will go and bind to the interleukin 2 receptor on the CD8 cell, and that CD8 cell will move toward the viral infected cells. All the cells of the human body contain the MHC class 1 molecule, and the immune cells that are patrolling in the body, they bind to the MHC class 1 molecule and recognize those cells as self cells of the own cells of the body. But if there is a viral infection in a cell, the viruses will be disintegrated by the lysozymes, and their antigens will be presented on the surface of the human cells along with the MHC class 1, and then the cytotoxic T cell that was activated by the helper T cell because of the interleukin 2 will bind to that MHC class 1 molecule along with which the antigen is present, and they will further activate the cytotoxic T cells, and the cytotoxic T cells will produce their cytotoxic effect, and they will kill that viral infected cell. So this was the response of the helper T cells toward the activation of the cytotoxic T cell and the killing of the viral infected cells. The other function of the helper T cell, the activated helper T cell, is the production of antibodies. How they perform this function? The helper T cells that were activated by the interleukin 2, they will further produce two types of interleukins that are involved in the activation of the B cells. First interleukin is the interleukin 4, and this interleukin is also known as B cell growth factor. So as the name implies, that it will cause the growth of the B cells. The B cells will proliferate and increase in number. The other interleukin that it produces is the interleukin 5, and it is also known as B cell differentiation factor. The B cells that are growing or proliferating, they will be further differentiated into plasma cells, and those plasma cells will produce antibodies. So this was an overview of the specific immune response and the arms of the immune response, such as the cell mediated immunity, in which the APCs and the T cell were involved, then the antibody mediated immunity, in which the helper T cell was activating the B cell and differentiating that B cell into the plasma cell, which was further producing antibodies. And the cell-mediated immunity includes the EPCs that presented antigen to the helper T cells, then the helper T cells further activated the cytotoxic T cells, and then the cytotoxic T cells killed the viral infected cells.